Hi gang! In this video we're going to talk about sampling, which I alluded to in the previous video, and in the second half of the video we'll talk about statistical inference. So to start us off, we're going to talk about different sampling methods and error associated with sampling. Of course, this PowerPoint is available to you on Canvas. Let's start off with a little bit of review. Generally, when there is a research question about a population, we need to collect a sample from that population, where a sample is just a subset of the population of interest. Once a sample is obtained, then we can collect information or data from the individuals in the sample. We called those observational units in the previous video. You can also call them experimental units if it's a part of an experimental study. The next several minutes are going to be dedicated to determining how is a sample actually collected. The gold standard to sampling is random sampling, and the go-to method of random sampling is what is called simple random sampling. Simple is the key word there. The criteria for a simple random sample to be collected is that, well, everyone has the same chance of being chosen. Makes sense, right? It's probably what you think about when you think of random. Furthermore, the individuals are selected independently. So whoever was chosen first is not going to have any bearing on who comes second. There is a technical note here that simple random sampling has more to do about groups being selected, but that's a technical note, and these 2 points that I have written here hold true. And we will use R during our lab days to simulate a randomly selected sample. So that's something to look forward to in the coming weeks. So simple random sampling, generally the go-to method for random sampling. There are other random sampling methods that we're going to discuss now. For one, we've got systematic random sampling. And this is when the individuals or the members are chosen at regular intervals. For instance, maybe every 17th individual is chosen, or every 5th person is chosen. Now, at first, that may not seem super random, but the place that randomness is inserted is where the starting point is. So if you've got a population, you wouldn't just want to start with a convenient location. You'd actually want to have a randomly chosen first individual and then select at random intervals. And how often do you select? Is it every 17th, every 71st individual? Well, if you just do this little division here, that should get you into the appropriate interval. Let's go to another random sampling method. This is called stratified. Stratified random sampling has a couple steps involved. If a population is present, first you divide the population into groups or stratified, or subpopulations. And everyone gets put into a group, and you can't have anyone existing in two groups at once. So that's what this non-overlapping means. Once you've got your population grouped up, then you take a simple random sample from each of these groups. There is a criteria involved with the creation of these groups. You'll notice in this image we've got little stars on top and then little squares, triangles. So the individuals in each of these groups is similar in some way. For instance, perhaps you are selecting from the HSU student population and you wanted to make sure that every major was represented in your sample. What you could do is divide up the majors. And now up here we've got the fisheries majors, and here we have the art majors and mathematics, etc. So each group is similar in some way, and then the sample is going to include a little bit from each group. So that's the benefit to stratified sampling, is that it will guarantee that each of those groups is represented in the sample. You can imagine if you really wanted each major to be represented in your sample, then this is a good approach to take, because you can imagine maybe you just did a simple random sample of students at HSU. It is possible you can select students randomly, but maybe a major was not included just by happen chance, not because of any nefarious or malicious intent. It could just be that no music majors were chosen. Another form of random sampling is called cluster random sampling. And this starts out in a very similar way to stratified, in that, again, you're going to divide up the population into groups, just like this little image shows. But this time, instead of selecting a little from each group, what is actually chosen is a simple random sample of the groups. So in this schematic here, this bubble, that entire group, gets put into the sample. And here, this little bubble, those 5 individuals all go into the sample as well. So here, these other 3 groups are completely excluded. So, it's cluster sampling. You divide up the population, similar as before. You take a sample of the groups, but then everyone in those groups is going to compose the sample. So this also has its benefits as well, depending on your particular study design. This could work well. Let's look at an example. We've got La Graciosa thistle, which is an endangered plant native to the Guadalupe Dunes on the central coast of California. In a seed germination study, 30 plants were randomly chosen from the population of plants on the dunes, and all seeds from the 30 plants were harvested. Okay, I want to give you a moment to consider which sampling method might this be. I may first consider, well, were there any groups happening? Because if there's some sort of grouping occurring, then we know we can whittle it down to either cluster or stratified. Careful consideration here is required. Notice that this is a seed germination study. So they are collecting seeds. To collect the seeds first, they are selecting plants. So they've got 30 plants that were randomly chosen. Certainly there's more plants out there on the dunes. But then all the seeds were harvested from those 30 plants. Here, this is a cluster sample because the grouping that was occurring was from the individual plants. So there were 30 plants that were chosen among all the plants, and those were the groups, and all the seeds in each of those groups composed the sample. So this is much like a cluster sample. Let's look at another one. In a study of parasitism of sand crabs, researchers obtain a random sample of crabs by dividing a beach into 5-meter strips parallel to the water's edge. These strips were chosen as the strata because crab parasite loads may differ systematically based on the distance to the water's edge, thus making the parasite load for crabs within each stratum more similar than the loads across the strata. Alright, so it kind of seems like there's some grouping going on here, right? The first stratum was a 5-meter strip of beach just under the water's edge, parallel to the shoreline. The second stratum was another 5-meter strip of beach just above the shoreline, followed by a third and a fourth, etc. Within each of these strata, 25 crabs were randomly sampled, yielding a total sample size of 100 crabs. So we've already given the hint. There's grouping going on, or strata. And now it's time to consider, is this cluster or stratified sampling? Because they are collecting individuals from each group, this must compose a stratified random sample. Contrary to our previous example with the thistle seeds, where here they were grouping across the plants, and they did not collect seeds from each plant. They collected all seeds in only 30 plants. So kind of a distinction there to consider. Let's talk about now issues with sampling. Because sampling is our go-to method to learn about populations without actually selecting an entire population, because we know there are limitations to that. Question: is a random sample going to look exactly like a population? Of course not. Because a sample, even if random, is only a subset of the population. It's probably not going to show all the diversity in that population. But this is to be expected. This discrepancy, if you will, is called random chance error due to sampling, or more simply, sampling error. So this is always present and should be expected. As long as a sample is randomly chosen, then any discrepancy between the sample and the population is due to what's called sampling error. However, if a sample is not chosen randomly, then this discrepancy may be due to bias. This is a 4-lettered word, the B word there. Let's talk a little bit about the B word, bias. Bias takes many forms. There are whole courses dedicated to appropriate for particular disciplines to minimize bias. So we're going to spend a couple of minutes on it, but certainly we can dive deep into this if this was a different course. But in general, bias is a systematic tendency for certain individuals to be selected more likely than others. This is bias, but in a statistical sampling sort of perspective. Here's an example of a potentially biased sample. A biologist plans to study the distribution of body length in a certain population of fish in the Chesapeake Bay. The sample will be collected using a fishing net. at first look seems reasonable, right? But this is going to lead to a biased sample because typically fish can slip through the holes in the net when the fish is smaller. So smaller fish are less likely to be caught than larger ones, and therefore that method of sampling would be biased. There's a lot more that we could discuss in bias, and there are several examples in your homework, so look forward to that. Let's talk now about statistical inference. To infer is generally to draw conclusions, and this needs to be based off of some sort of procedure in statistics. Let's talk about that. And well, here's the hint. It's all about randomization. Learning about a population achieved through sampling. Learning about the sample in and of itself is of value and sometimes all that is of interest. However, typically, we hope as researchers to learn about a sample so that we can actually learn about the individuals in the population, those who were not in the sample for the larger group. In order to do that, to extrapolate, if you will, there's a lot of statistical theory beyond the scope of our class that allows for this generalization or extrapolation beyond the individual observations that were observed to a broader context. So, no such statement or generalization can be made with absolute certainty. Therefore, every such generalized statement should include some measure of uncertainty. This is because even random samples are subject to sampling error, and that can lead to a sample that is not representative of the population. Even though the sample came from the population, it may not truly reflect the population. And so a question worth addressing is how representative is a sample likely to be of its population? Now, these are pretty jumbo ideas for the first week of class, and so we're going to unpack these topics over the course of this semester. Let's take a look at how sampling and randomization in particular may affect the type of inference that can be made. In general, there are two types of statistical inference. One of them is called population inference, and that is when you extrapolate or generalize from your sample to the population. And then there's a causal inference, i.e. cause and effect. These types of inferences do not come for free, but depend on some sort of randomization feature of the study design. So it is the random selection of individuals that allows for an inference to the population, and it is the random allocation or distribution of units to treatments that allow for a cause and effect inference to be discovered. Let's look at both of these in a bit more detail now. So how does random sampling allow for an inference to the population to be made? That's because random sampling is sort of I call it our insurance policy. that allows us to assume that the sample was representative of the population. That's not always true, but in the long run, random sampling does allow for a population to be fairly represented by its random sample. If what you observe in a sample is a fair representation of what's going on in the population, then it seems reasonable then to extrapolate or generalize what was observed in the sample is happening in the population. Now, it's very important then that you have a well-defined population. Particularly, you want to make sure kind of all facets are covered, particularly space and time. For instance, information collected in 2009 may no longer be applicable in 2020 because things change over time. And that may even be true on a shorter time scale of seasonality, spring versus fall. Also, space. For instance, the Puget Sound orcas are probably exhibiting different behaviors and social structures than the orcas in the Southern Ocean. So time and space are important considerations when defining a population. An inference cannot be made to a population that was not represented in a sample. Let's look at the other type of statistical inference. This is cause and effect, or causal inference. And this applies to experimental studies because this is where there are treatments and the individuals are randomly assigned to those treatments. If that is achieved, then the cause and effect inference can possibly be made. Why? This is because by randomly allocating individuals to treatments, this ensures that subjects with different features are mixed across the treatment groups. For instance, if you had two treatment groups, in the previous video we said red pill, blue pill, you wouldn't want only males in the blue pill group and only females in the red pill group. You would want a mixture of both sexes in both groups. If you had not mixed the groups, you don't know what was the response due to. Was it due to the pill or was it due to the sex? So this randomization of subjects across the treatments, so any differences across the groups are not due to some systematic procedure which could potentially be biased. This ultimately reduces the effect of confounding variables. It is those confounding variables that in an experiment, researchers are trying to minimize the effect of those confounding variables. There are a couple other points here for a cause and effect or causal inference to be valid. Well, it's an experiment, so there has to be treatments to compare. Usually there is a control group or a placebo group in addition to any other group or groups. Furthermore, there must be some sort of replication. There can't just be one subject in each group. There needs to be more than one subject for each group. That's the property of replication. I have a question for you. Can a causal inference be drawn from an observational study? I'll let you think about that. Let's look at an example of a study and we will dissect some of its properties. Researchers used 7 red and 7 black playing cards to randomly assign 14 volunteer males with high blood pressure to one of 2 diets for 4 weeks. One of them got a fish oil diet, and the other got a standard oil diet, whatever that means. They were measuring the reduction in diastolic blood pressure in each subject. I've even got a copy-paste image here of some of the data. So we have the reduction of the 7 individuals in the fish oil diet, and then the 7 individuals in the other diet. Now this one, there's a couple negative signs here. Because this is measuring the reduction in blood pressure, because there's a negative sign on some of these, those individuals actually had an increase in diastolic blood pressure. Okay, I've got some questions for you about this study design. Do the volunteers constitute a random sample? The answer is no. They volunteered. They were not randomly chosen. Okay, so therefore, any results observed in this study only applies to these 14 individuals. Why might the results of this study be important even though the volunteers do not constitute a random sample from any population? Well, the conclusion that the fish oil diet causes a reduction in blood pressure is still a useful one, even if it only applies to these particular individuals. Now, why can we say in this scenario, why does it appear that fish oil consumption causes a reduction in blood pressure? That's because the 7 people in each group were randomly assigned to the 2 diets. By doing so, they were able to ensure that each diet contained a mixture of elements present in these 14 volunteers. Cool. I have one more point to make, and this is regarding terminology, the terms sample versus specimen. The word sample is used a lot in our statistics course, but it usually means something different in other disciplines, such as biology. For instance, if a biologist collects blood from 20 people and measures anything, glucose, they might say that they have 20 samples of blood. 20 people, 20 samples of blood. However, the statistician, which is you, would say that you have 1 sample of 20 glucose measurements, and the sample size is 20. So in the interest of clarity, let's try to use the word specimen where a biologist might prefer sample. Well, that's it for us. What should you work on in the near future? Make sure to complete the upcoming Canvas quizzes and start the first lab assignment. I highly recommend reading the first chapter in the textbook if you would like more examples of sampling methods and study design. Have a good one.